Well, aloha everyone. Welcome back to Human Nutrition here at Chaminade University. Today we will be delving into Unit 6, which is going to cover proteins. We'll be describing proteins, the basic structure of a protein and multiple shapes that proteins can take, and also the classifications of amino acids, which are the monomers of which proteins are constructed. We'll talk about protein digestion and absorption, and we'll talk about how we metabolize amino acids and how we use amino acids within our body. We'll talk about the functions of protein within the body and also talk about the daily recommended amount of protein that you're supposed to ingest based on the DRIs. We'll also talk about food sources that provide protein, which are better or worse for you, and the methods available to determine the protein quality. And then we'll talk about the health consequences of overconsuming or underconsuming protein and the benefits and risks of a vegetarian diet. So what are proteins? Proteins are a macromolecule. They're nutrients found in every cell in the body, and they participate in almost all cellular activities. All enzymes are made of proteins. When we talk about catalyzing cellular reactions, those are enzymes. Most hormones are made of proteins, and almost all of the cellular activities and responsibilities are performed by proteins. Now, proteins are biomolecules that are made up of monomers of amino acids. So they're long chains of amino acids that are made based on the DNA. So basically DNA creates RNA and RNA creates protein. Now on the left we have different types of macronutrients. Here's carbohydrates, lipids, and proteins. And again, each of these are going to have monomeric and dimeric and polymeric forms. Monomerics being the smallest train car that you can break it down into that still retains the properties. And if you put several of those train cars together, you get a dimer, trimer, etc. until you finally get a polymer. And for proteins, the monomeric form is amino acids, and the polymer would be a peptide, or eventually a long polymer would be a protein. Now, if we eat too much protein, we can't store it in the body. So excess protein is actually just going to get secreted out in our bodily wastes. Now, proteins contain nitrogen as well as carbon, hydrogen, and oxygen, and some proteins contain sulfur, which is not found in carbohydrates or lipids. It's a very specialty. It gives a protein a very specialty function in that some proteins are able to make what's called disulfide bonds or cytoplasmic bridges, and these guys basically cysteine bridges, and these are basically going to be like Velcro ways in which the protein is going to be able to undergo secondary or tertiary structure, whereby amino acids that aren't anywhere near each other in the sequence are able to come together when the protein folds and actually adhere to one another. And again, these are called cysteine bridges or disulfide bonds, and it allows the protein to fold on itself. Now, proteins are comprised of amino acids. We have 20 amino acids that are used to make proteins. And proteins are classified multiple ways, but one of the ways they classify them is by the number of amino acids that are found in the chain. So if we have less than 50 amino acids, that's called a peptide. And if we get really small, just 2 amino acids, that's called a dipeptide, 3 tripeptide, and anywhere that's over 10 would be called a polypeptide. After we hit the 50 amino acid length, then we're going to have called this a protein. Typically, proteins are approximately 100 to 10,000 amino acids linked together. So these can be extremely long, and they take a bunch of different structures and shapes. So this is a monomer of a protein. This is called an amino acid. An amino acid is called such because it has an amine group on one side and a carboxylic acid group on the other side. Additionally, so there's a carbon in the middle with the amine group, the carboxylic acid, and a hydrogen. This is going to be the same for all of the amino acids. But what's going to change is this side chain region. So we can have something that attaches as the fourth connection to the carbon that's going to confer specialty ability to that amino acid. So different amino acids have unique side chains. And I won't ever ask you to draw these out, but I want you to understand that some are going to be very large, some are going to be very small, some are going to be charged, some are not going to be charged. And so the size and the charge is going to determine the function of that amino acid within the protein structure. Again, amino acids have one side that is going to have an amine group, another side that has a carboxylic acid group, then a hydrogen, and then a side chain that's going to be variable. And this variability of these side chains is going to give unique properties to each amino acid that is going to allow it to have its own unique functions. Now, when we put two amino acids together, that's going to create a peptide bond. The carbon of one amino acid on the acidic side is going to join with the nitrogen of another amino acid on the amine side. Now, if you look, this amine cannot bind to this carboxylic acid. So within the same amino acid, this bond is never going to happen. This bond is going to happen between an amine group of this amino acid and a carboxylic acid of another amino acid, or of this carboxylic acid and an amine group of another amino acid. And the peptide bond is formed like all bonds through the process of condensation or dehydration synthesis, and broken through the process of hydrolysis. Here's an example of what I mean by that. So here we're going to have two monomers, right? So that's two amino acids, and they're going to come together by dehydration synthesis or condensation reaction, whereby we're going to remove this OH and this hydrogen group, one from this amino acid and one from that amino acid. Those two come together to form water and is going to confer a peptide bond between this carbon and that nitrogen. That here's the peptide bond that holds these amino acids together. And when we get a long chain of them, we'll have multiple peptide bonds. Now, if we were to break this dipeptide into its monopeptides or its amino acids, that would be the process of hydrolysis. Again, we would split a water molecule into the OH and the H groups, and that would cap the sticky ends of each of the monomers, allowing them to become their own individual amino acids. So creation of a peptide bond happens through condensation, and the breakdown of that peptide bond happens through the process of hydrolysis. Now, we have essential and non-essential amino acids. All essential amino acids are considered essential because we have to consume them in the diet. We're unable to create them ourselves. And there are 9 amino acids that are considered essential. And then we have to get them from exogenous sources. And then we also have non-essential amino acids, which are amino acids that we can synthesize in our own body. We have 11 of those. Now, some of these non-essential amino acids are considered conditionally essential, in that usually they're non-essential, but sometimes the body cannot make enough of them. So therefore, they would become essential. Some examples of this would be tyrosine and glycine. Generally considered non-essential, but in certain situations, you're going to actually have to get them from exogenous sources because you can't keep up with their creation. Now, I'm not going to ask you to know which ones are essential and which ones are non-essential, but this here is a list of the 9 essential amino acids and the 11 non-essential amino acids, or conditionally essential amino acids are denoted in italics. As I mentioned, proteins have multiple different levels of structure. And if you're looking at just the amino acid sequence, what order the amino acids are going to be created or linked together in, that's called the primary structure. But we have a secondary structure, which is the 3-dimensional structure of the protein that happens as it folds and twists during creation. And then a tertiary structure would be the final 3-dimensional globular structure of the protein itself. And if we have more than one protein coming together, so 2 or more polypeptide chains bound together, that can make the quaternary structure of a protein. What do I mean by that? All right, so if we're just going to talk about how the amino acids are bound to one another, in what order, via the peptide bonds, that's considered the primary structure, A, B, C, etc. But there are other ways that these proteins can fold, making secondary structures such as alpha helices and beta-pleated sheets. So if you were to picture a spiral staircase and everyone's standing on the spiral staircase, you could potentially hold hands not only with the person in front and behind you, but also with the people on the stairway above and below you. So that would allow you to make bonds, say, between this one and that amino acid, right? And that is going to allow us to make what we call secondary structures. Some common secondary structures include alpha helix, you could picture that like the spiral staircase, and beta-pleated sheets, so you can picture that like the corrugation in cardboard. And when we finally make the final structure of that particular series of amino acids, that's going to be the tertiary structure. And if multiple peptides come together, as in the case here of this hemoglobin molecule, that's going to be the quaternary structure. So we have 4 independent globular molecules to make this hemoglobin molecule. All right. So, as I mentioned, proteins are folded as they are created. And that folding is very specific, and it can actually be undone. So if we destroy the protein by unfolding it, that's going to be permanent, and that's called denaturation. And we can denature proteins in multiple ways: high heat, extreme pH, high or low, so acids or bases, and high levels of salt, and also physical mechanical agitation. And when we denature or alter the shape of the protein, that alters its function. And when we entirely denature it, we get down to its primary structure. So the primary structure remains unchanged, but we've lost all of our secondary, tertiary, and quaternary structures, which renders the protein essentially useless. So this is what happens when you denature a protein. If you figure that this 3-dimensional structure is like having a crumpled piece of paper, when you denature it, it's like taking an iron and flattening that piece of paper back out. And try as you might, it's impossible to crumple that piece of paper into the exact same ball that it was crumpled into before you ironed it. So we're never going to be able to get back to this normal protein shape or normal protein function after we denature the protein. So while we are consuming protein, how does digestion occur? So the first time that we encounter protein digestion is when the bolus of food enters the stomach. As we know, gastrin is going to stimulate the release of HCl from those gastric pits, and HCl denatures the protein and does a secondary thing. It breaks down something that's a precursor molecule of pepsin. So pepsinogen is the precursor molecule of pepsin, which is an enzyme that breaks down protein. Now, pepsin is a specific protein that works only in low pH in the environment of the stomach, and its responsibility is to break down polypeptides into shorter chains. So we're going to start with long proteins and break them down into medium-sized chains. And digestion is going to continue in the small intestines, where we're going to encounter several different types of peptidases. So polypeptides, which are now going to be smaller versions of that larger protein, enter into the small intestines, and they're going to be broken down into smaller peptides. I want to take a moment to digress here and mention that in the small intestines, we are restoring pH to a more neutral, somewhere around 6.5 or 7, and this is going to be the pH by which most of these enzymes are going to function. So at this point, pepsin, which just came from the stomach and works at really low pH, is going to end up out of the pH range for it to function. So although it's still present, it won't be functioning as a digestion molecule in the small intestines. Now we have proteases that are released by the pancreas, and they're stimulated by CCK. We also have proteases, again, these are released by the pancreas, that are responsible for breaking down polypeptides into tripeptides and dipeptides, as well as proteases such as dipeptidases and tripeptidases, which break down dipeptides and tripeptides respectively into their amino acids. So we're breaking down large series of proteins, right? Large polypeptides into shorter chains in the stomach. Those shorter chains in the intestines are going to be broken down by proteases into di and tripeptides. And those di and tripeptides are broken down by dipeptidases and tripeptidases into their amino acids. And again, that occurs in the small intestines. So this is a summary of the different digestive enzymes that are involved, including pepsinogen, which is going to become pepsin and is going activated to pepsin by HCl and be responsible for breaking polypeptides into shorter polypeptide chains. So large polypeptides to small polypeptides. Trypsin in the small intestines. Remember, pepsin only works at low pH. It will not work once we enter into the small intestines. But trypsin and chymotrypsin do, and they come from the pancreas as well as carboxypeptidase, and these are going to help break down peptide bonds. And carboxypeptidase is going to break one amino acid off from the carboxyl end of a peptide chain at a time. So that's actually going to be releasing one amino acid. We also have aminopeptidase, which breaks the free end of the amino acids from the tri and dipeptides into single amino acids. Tripeptidases and dipeptidases. So aminopeptidases, tripeptidase, and dipeptidase all come from the brush borders of the small intestines and are responsible for breaking down the amino acids into their single amino acid components from larger peptides, including tri and dipeptides. So how are they absorbed? They're absorbed directly across the small intestines and transported to the liver via the portal vein. And in the liver, they're going to have multiple different functions. Mainly, they're going to be used to synthesize new proteins. But if we're low on energy, we can convert them into glucose or energy. We can also convert them into fat if needed. And then they can also be released in the bloodstream and transported to other cells throughout the body where they will be used to synthesize new proteins. Now, occasionally proteins are absorbed intact or close to whole, but this is very unusual because if you're eating protein from another animal, it's going to be a cow protein or a pig protein. It's not going to be a human protein that we're able to use immediately. So we're going to have to break them down, generally break them down into their amino acids so that we can then rebuild them into human proteins. So where does that happen? Again, we're going to skip the mouth because we don't have any peptidases in the mouth. First time we see our peptidases are going to be in the stomach. The first one we're going to interact with is pepsin, which is broken down from pepsinogen in the stomach under low pH conditions. So that's going to start breaking down large proteins into smaller polypeptides, and those smaller polypeptides enter into the large intestines, where trypsin and chymotrypsin take over, and they're going to break down the smaller polypeptides into di and tripeptides, as well as some free amino acids. And that's where we're going to see absorption as well as in the small intestines. Now again, after they travel out of the small intestines, they're going to go through the portal vein into the liver, where they're going to end up being metabolized. And this is going to be based on the needs of the body. So if we need more or less of particular proteins, we're going to be creating those proteins from those amino acids. Now, most amino acids are actually going to be sent into circulation through the bloodstream, where they're going to be picked up by cells and used to create proteins within the cell. However, sometimes if we're low in sugars, so if we're not eating enough carbohydrates, the amino acids can be converted into glucose, which can be used as an energy source. Now, the body is constantly creating new proteins, and it's going to need to use amino acids to do this. So we're going to constantly need an amino acid supply to be able to create new proteins, so for protein synthesis to occur. So the body is constantly breaking down proteins and building up proteins daily. This allows us to maintain body structures such as our tissues, our cells, our organs, and we use an amino acid pool to be able to have a supply of amino acids readily available for the body to be able to create whatever protein it needs on demand. Now, if a person is extensively injured, they might need extra protein for healing instead of just for maintenance. And all individuals have what's called protein turnover, or the degradation and resynthesis of proteins. In fact, in a normal individual, over 200 grams of protein is turned over every day, broken down, and resynthesized. So this demonstrates the metabolic fate of the amino acids. So amino acids can either get sent for protein turnover, and this is what they're usually used for, the creation of new proteins. But they can also be used for gluconeogenesis, or the creation of sugars, if we are limited in sugar in our diet. They can also be used to create ATP, or when their diet is deficient in kilocalories, so we can burn them immediately for energy, or we can store them as fat by converting them into fatty acids and stored as triglycerides in the adipose tissue. And this happens when we have excessive kilocalorie intake. So depending on what the body needs at any given moment, we can either use amino acids to make proteins, to make glucose, for ATP, or for energy creation, basically, and also for the storage within the fat cells in the form of triglycerides. Okay, so how are proteins made? There are three major stages of protein synthesis. And the first happens in the nucleus of the cell. That's called transcription. And transcription is when we're taking DNA and we're making a copy of it in terms of making messenger RNA. And it's actually an inverse copy of it. So if you had a black and white book, you would now end up with a white and black book. So it's going to be an inverse copy of the DNA. And we'll talk about that. I guess you might talk about that in other courses. But basically what happens is that's going to leave the nucleus and enter into the cytoplasm, where it's going to undergo the process of translation. Essentially, mRNA is going to bind to that ribosome and then read the codon sequence. And every codon, or set of 3 nucleic acids, is going to cause a transfer RNA to bind and bring one amino acid in based on whatever the sequence is in that code. And this process is going to continue, called elongation, as amino acids are brought in one at a time by one tRNA molecule to be able to build a chain of the appropriate sequence, which is going to happen based on what it's reading on the code in the RNA. And that's going to continue until the sequence is finished and we end up hitting a stop codon or a release codon, and the protein is released. Now, if we have abnormal protein DNA, or if we have abnormal DNA that codes for a particular protein, then we're going to end up with an abnormal protein at the end. Abnormal DNA makes abnormal RNA, makes abnormal protein. And an example of a disorder that is created by abnormal protein would be sickle cell anemia. It's a fairly common blood disorder whereby the blood cells end up sickle-shaped instead of torus-shaped or dimpled. And that means that we're going to have abnormal proteins, which means that those abnormal proteins, or abnormal hemoglobin, are going to be unable to carry the same amount of oxygen as the normal proteins. This is an overview of protein synthesis. So again, DNA makes RNA and RNA makes protein. DNA and RNA differ in many different ways, but some of the major differences is that DNA is double-stranded and extremely long. I like to use the example of making a cherry pie, which would be the protein. Now, in order to make a cherry pie, we need a cherry pie recipe, and that would be the mRNA, right? So you're going to have to figure out what you're going to make based on the recipe. And in order to get the recipe, you'll have to go to a recipe book, and that would be considered like the DNA, where we have recipes not just for the cherry pie, but for all kinds of desserts and perhaps all kinds of dinners, etc. So a chromosome, which is comprised of DNA, is extremely long, and it shares many different genes. So it's going to be a very large recipe book that has a ton of different recipes on it. However, each RNA is only going to be a Xerox of a particular recipe. So the RNA is only going to be a copy of one gene, not of the entire chromosome. And once that copy is created, it's going to exit the nucleus. It does that by way of the nuclear pores. And there's actually little gate guards here that are going to determine whether or not this is RNA or DNA. One of the things I forgot to mention is that one of the other major differences between RNA and DNA is the type of sugar that's used. RNA uses a sugar called ribose, and DNA uses a sugar called deoxyribose. right? So it has one less oxygen. And these gate guards at the nuclear pores are able to determine if it's ribose or if it's deoxyribose. And if it's ribose, it'll be allowed out. If it's deoxyribose, it will not. And in this way, the RNA is going to leave the nuclear pore where it's going to encounter ribosomes that are either going to be free in solution or bound on this rough endoplasmic reticulum. Either way, the role of the ribosome is to read this transcript. These are the cooks that are going to read the recipe and create the cherry pie. And they do that by shuffling along three what's called nucleotides. Each set of 3 nucleotides is called a codon, and you can see each codon is going to have what's called an anticodon. So then when I said an inverse section, this anticodon is going to be attached to DNA, or sorry, to tRNA, I apologize. So this tRNA molecule is going to bring in one amino acid, and each tRNA is going to be specific to the amino acid that it carries, as well as the anticodon on it. And when that anticodon matches the codon sequence from the RNA, now that amino acid is going to get transferred to that growing polypeptide chain through the process of elongation. And eventually we reach a stop codon where the ribosome will fall apart, and it can either start back at the beginning and read this transcript again, or it can find another RNA transcript, but it will continuously create more and more protein until it reaches its half-life. All right, so that's the overview. We have transcription occurring inside the nucleus, exiting out through the nuclear pores in the form of RNA. RNA is then going to undergo translation, which happens in the cytoplasm, which is the creation of proteins, as it's read by ribosomes using tRNA, or transfer RNA, with its anticodon-codon matching, which brings in one amino acid at a time to the elongating polypeptide chain. So I know that seems like a lot, and I did kind of brush over it, but that's kind of the overview of what happens in your cell. So what happens when we have enough amino acids in our body to be able to meet the needs of protein creation? Well, that's called capacity. And once the amino acid pool reaches capacity, the amino acids are going to be broken down into their component parts and used for other things. And that means that we're going to undergo a process called deamination. And when we undergo deamination, it's exactly what it sounds like. It's the removal of the amine group. So when the amine group is taken off from the amino acid, ammonia is formed, which is converted to urea in the liver and subsequently excreted in the urine. So if you're looking at urinalysis and you see high urea levels, it's probable that we are undergoing the process of breaking down or deaminating our amino acids. What's left? What's left is these carbon-containing remnants, and those are going to be converted into glucose, if possible, through gluconeogenesis. Now, only some amino acids lend themselves to that. Those are called glucogenic amino acids, but if they are glucogenic amino acids, we can create glucose through the process of gluconeogenesis. Otherwise, they can be converted to fatty acids and stored in adipose tissue, or oxidized through metabolic pathways and turned into energy immediately. Okay, so we talked about deamination, which is taking the amino group off of an amino acid. But let's talk about transamination, which is taking the amino acid off, taking the amine group off of one amino acid and moving it over to another amino acid. In this way, we can take non-essential amino acids. We can create non-essential amino acids from other amino acids. So we take the nitrogen from amino acid A and move it over to a different amino acid, specifically a keto acid. That's going to make a new non-essential amino acid. So we're able to create amino acids from other amino acids. So if we have too much of amino acid A and we need more of amino acid B, and both of them are non-essential, then we're able to do that by the process of transamination. Okay. Again, we can also use protein for gluconeogenesis. That means that we're able to create new, so genesis, creation, new glucose molecules. So the creation of new glucose molecules from proteins. And this can happen if we're lacking carbohydrates in our diet. If we're low on carbohydrates, the body is going to convert amino acids, particularly glucogenic amino acids, into glucose. And if we're eating too much protein, so excess protein in our diet, it can be converted again to fatty acids and stored as triglycerides in our adipose tissue. But if too few kilocalories are consumed, that protein will not be stored as fatty acids in the adipose tissue. Instead, it'll be oxidized and used for energy as an immediate energy source. All right, so what do proteins do within the body? They do a lot of things. Pretty much all of the functional roles within the body are performed by proteins. Proteins are structural support. They also allow the cells to move around. They act as biological catalysts to speed up cellular reactions. They can also act as chemical messengers in order to send information into the cell, basically knocking on the door and sending an intracellular cascade within the cell. Things like hormones are a good example of this. They're involved in regulating homeostasis of all things, including fluid balance, pH balance, acid-base balance, salinity, etc. So proteins play multiple functions within the body. They also transport substances from one location to another, including oxygen, waste products, lipids, vitamins, salts, potassium, etc., through the blood and through the cell membranes. And they're responsible for your immune system. So, antibodies are something that we may have talked about previously, but they're going to be involved in the immune system and the immune response. And antibodies are also proteins. They're specialty proteins that are responsible for binding to pathogens and then neutralizing them so that our body can remain unscathed. Additionally, we can use them to provide energy. Again, they're going to provide about 4 kcal per gram. And if you eat more proteins, you can improve what's called your satiety, which is your feeling of being full. And so they can help with appetite control as well for people who have issues with overeating. This diagram depicts enzymes. So enzymes are biological catalysts. They're going to speed up the rate of the reaction. Here we have an enzyme with its active site, and the active site is where our substrate is going to bind. A substrate is just whatever compound we're going to be converting. We can take one compound and break it down into two, or an alternative reaction would be to take these two products, put them in here, and create this compound. So enzymes are going to mediate only one specific reaction, but there is an equal and opposite enzyme for almost every reaction. So this enzyme is going to take compound A and break it down into B and C. Enzymes remain unchanged at the end of the reaction, but throughout the reaction, they do undergo a shape change. So basically what happens is the compound is going to bind into the enzyme, and then the enzyme is going to undergo what's called a conformational shape. And that means that it's going to force a change in the compound that's locked in that active site. Once that change has occurred, the products no longer fit in the active site and are going to pop out. Once the products are released, the enzyme is available for another reaction, and it's just going to lather, rinse, repeat until it reaches a state of degradation. Proteins can also transport things into and out of the cell. Here's an example of the sodium-potassium pump, whereby sodium is going to bind to the inside of the transport protein on the inside of the cell. Sodium gets pumped outward, and potassium is going to bind to the inside of this protein from the outside of the cell, right? So once potassium binds, then the conformational shape takes place, and the protein is going to release the potassium inside of the cell. In this way, sodium can be pumped out and potassium pumped in, making a constant gradient, allowing us to bring things back in via the process of symporting once sodium wants to come back into the cell. So what do proteins do? All sorts of things. Structural proteins, mechanical support proteins, maintenance proteins. Proteins are enzymes. Proteins are hormones. Proteins are involved in homeostasis, including the regulation of fluid balance, acid-base balance. Proteins transport things, the oxygen, waste products, etc., throughout the blood into and out of the cells. Also involved in the immune response, such as antibodies. They're involved in energy, so we can use them to create energy if we need to. And they're also involved in the ability to control your weight gain by the control of your appetite. So how much protein do we need to eat daily? So we want to make sure that we are in what's called nitrogen balance. And again, there are nitrogens found in proteins. And you're supposed to consume enough to replace what you use. That means that we should be in stationary nitrogen balance. Now, some people are in what's called positive balance, which means that they are consuming more every day, and that's because they are actually going to be either creating new tissue like pregnant women or people recovering from surgery or growing children. Now, some individuals have negative nitrogen balance. For example, these are individuals that would be fighting an infection or coming out of surgery or suffering from severe emotional trauma. And what do I mean when I say in nitrogen balance versus positive or negative nitrogen balance? So in an equilibrium, you would have the same amount of nitrogen coming in as excreted. And this is where a healthy adult typically tends to fall. Now, if you're an unhealthy adult, someone who has just experienced a medical trauma or is not eating a healthy diet, you might be in negative balance, meaning that you're not taking in as much as you're excreting. So you're actually going to be deficient in nitrogen. Sometimes people are taking in more than they are excreting, such as pregnant women or growing children and adolescents, some athletes, people who are building tissue. They tend to be bringing in more nitrogen than they're excreting because they're storing that in the creation of new proteins. So this is an overview of nitrogen balance versus reasons why it might be out of balance. Now, your daily recommended protein intake is approximately 0.8 grams per kilogram daily for adults over 18. And we should be consuming approximately 10 to 35% of our daily kcals from proteins. Now, what's also true, though, is that individuals who are overweight might have different needs than normal weight individuals of similar height, simply because they have extra to begin with. Now, the College of Sports Medicine, Academy of Nutrition and Dietetics, and other experts advocate that approximately 50 to 100% more protein should be consumed by competitive athletes, especially if they're performing endurance exercises or resistance exercise. Now, typically this population is already eating more, and so they're already getting additional protein into their diet. However, not all protein is created equal. We have high and low quality protein. Now, in order for protein to be considered high quality, it has to be digestible and have all of your essential amino acids, plus it has to provide sufficient protein to synthesize all of your non-essential amino acids. And we have several ways that we are able to determine the protein quality of foods. We use what's called the amino acid score, which is the composition of all of the essential amino acids in the protein compared to a standard, and that standard is usually albumin, which comes from egg protein. We also have PDCAAS, the Protein Digestibility Corrected Amino Acid Score. And this includes the digestibility of protein as well as the amino acid score. So basically, how much of the protein are you actually going to be absorbing based on how much, compared to how much you're actually ingesting. And we use this to determine the percentage daily value of proteins in a serving of food. And lastly, we also have the biological value, which is going to be how quickly the nitrogen can be synthesized into body protein. So you're going to ingest that nitrogen and absorb protein. How quickly are we able to then convert it into usable proteins for the body? Now, we also can split proteins into complementary or complete or incomplete. Now, complete proteins are going to have all 9 of the essential amino acids. There's nothing lacking. If you were to eat just this protein, you could hit all of the amino acids that you need that are essential, meaning that you have to get them from exogenous sources. Most animal sources are complete proteins. Now, soy protein is also a complete protein that doesn't come from animal sources, but it's going to be the exception. Plant proteins typically are going to be incomplete proteins. So typically complete proteins are considered higher quality than incomplete proteins, and an incomplete protein is one that's going to be low in some of the essential amino acids. That's going to be the limiting amino acid of this incomplete protein, and usually, again, this comes from plant proteins. So this is why vegetarians have to be very careful when they are planning their diet that they're going to consume what's called complementary proteins. One incomplete protein from food source A and another incomplete protein from food source B that together supply the limited amino acids. So if incomplete protein A is missing amino acids A and B, but incomplete protein B is missing amino acids C and D, then we're going to be able to have complementary, because A is going to provide what B is lacking, and B is going to provide what A is lacking. So a combination, for example, of greens and legumes will give you complementary proteins. Even though both of them are incomplete, consuming both of them together will complete what you need in terms of your essential amino acid consumption. Now, many places that contain protein, many types of food that contain protein include eggs, meat, fish, soy, and dairy. And if we have a 3-ounce serving of meat, poultry, or fish, that's going to provide between 21 and 25 grams of protein, or approximately 7 grams of protein per ounce. And size-wise, you're looking at something about the size of a deck of cards. And this is adequate amount for one meal. This is an adequate serving of protein for one meal. So oftentimes individuals overeat their protein, because when you get a burger, for example, it's never 3 ounces, so typically 5 or 6 ounces. Sometimes you get a serving of food at a restaurant, that'll be a 16-ounce steak, right? And that's way more than what you need for one individual serving of protein. Now, typically, individuals, particularly in the United States, tend to get enough protein in their diet, so taking protein supplements is typically unnecessary and not necessarily recommended unless you intend to be a bodybuilder who is bulking up and needs weight gain or something like that. This diagram depicts major food sources of protein from your vegetables. We get a good bit of protein, your grains. We are going to get the highest amount, of course, from our meats, fish, meat, poultry, etc. But we're also going to get some protein from our dairy, as well as our nuts and our grain sources. Okay, so what happens if you overeat or undereat protein? So if you eat too much protein, that can increase your risk for heart disease and for kidney stones. If you intake protein with high saturated fats, you can end up with an increased risk for heart disease. So you want to make sure that you choose a variety of plant proteins to help reduce the risk of heart disease. Additionally, a diet that's high in animal protein and also low in carbohydrates can change the pH of your urine, when a low urine pH can raise your risk of developing kidney stones. So if you have too much protein in your diet, it can increase your risk for heart and kidney disease and disorders. Additionally, it can increase your risk for osteoporosis, because if we eat more protein and less calcium, that can lead to increased urinary calcium losses. However, if we have a high protein diet with adequate calcium, especially from dairy sources, this can help protect our bones. So getting protein that includes calcium in it can help protect us. And if we have too low a protein intake, this can lead to bone loss, especially in elderly men and women. It can lead to osteoporotic bone lesions. Now, if we add too much emphasis on protein in our diet, we can tend to displace other important food sources such as whole grains, fruits, and vegetables. On the flip side, if we ingest too little protein, we can end up with protein energy malnutrition. And that means that we can start using proteins for energy sources rather than for its other functions in the body, which can be deleterious to the individual because you're removing the protein from regions of the body that need it for functions. And typically this happens when other important nutrients are also in short supply. This is something that can be more prevalent in infants and young children, as they're actually going to be developing more and more cells every day, so they have high protein and energy needs. Now, if we end up with eating inadequate protein in our diet for longer periods of time, we can end up with digestive tract disorders, because the cells lining the digestive tract can be slowly sloughed off without being replaced, which can inhibit digestive function, which of course can inhibit absorption of food, and can also mean that intestinal bacteria can get across that GI tract and into the blood, which causes sepsis or septicemia. Can also lead to immune system compromisation, because malnutrition can lead to the inability to fight infection. Again, your antibodies are proteins, and if we're using that protein for energy sources instead of for its functions, then we can end up with a compromised immune system. If we eat too little protein, again, that's PEM, this is not something that the developing world sees very much, but individuals in areas of the world that are third world nations, especially children, end up with a problem with protein intake. Approximately 1 in 8 people in the world have low protein intake or low kcal intake, or both. And that means that the available protein in their bodies is used as an energy source rather than for the roles of the body that they would normally play, which means that that lack of protein can cause issues with the GI tract and inhibit the ability of those individuals to absorb nutrients. That means these individuals typically are going to have compromised immune systems as well. And we have 3 major types of protein energy malnutrition. The first is called Kwashiorkor. The next is called Marasmus, and we also have Marasmic Kwashiorkor. Now, Kwashiorkor is a severe protein deficiency, which is generally the result of a diet high in grains but deficient in protein. So a lot of the world subsides almost entirely on rice or on grains. Some symptoms of this severe protein deficiency include edema in the legs, feet, and stomach. So individuals would often be seen as having eaten sufficient food, even though they are starving, because they have this bloating of the stomach. Also leads to diminished muscle tone and strength, and brittle hair, which is very fragile and easy to pull out. Remember, hair is going to be comprised of keratin, which is a protein, and if proteins are being absorbed for energy, they're not going to be put into the hair. It can tend to lead to a pale, sad, and apathetic appearance, and a decreased immune response. So these individuals end up being prone to infections, septicemia, and also have issues with water and electrolyte imbalances. Now, by contrast, here's Marasmus. We're still going to have a severe deficiency in kilocalories as well as proteins, leading to a pale and emaciated appearance. They end up weakened and appear very apathetic. Often they're unable to stand without support because their bones are so brittle, plus they have very little muscle tone. So no muscle tone means we're not able to support our own weight, typically appearing very old, and again with the thin hair that's very brittle. They have low body temperatures and low blood pressure and are prone to dehydration, again infections, and unnecessary blood clotting. Now, Marasmic Kwashiorkor is when we have a chronic deficiency in both kcals and proteins, leading to, again, edema in the legs and arms, skin and bone appearance. And these individuals are able to be treated whereby we'll end up with a subsidation of the edema, so the edema will lessen, and that will mean their appearance will become somewhat more like Marasmus, which is here. Okay, so if we're deficient in proteins, we get Kwashiorkor. If we're deficient in kilocalories, we get Marasmus, and if we're deficient in both, we end up with Marasmic Kwashiorkor. So how do we treat these individuals? So we're able to offer medical treatment and nutritional treatment, which can dramatically reduce the mortality rate of these individuals. There's a couple different steps we want to take, though. First, we want to address the life-threatening factors like dehydration and fluid imbalances. So we want to make sure that we're not just going to immediately feed these guys a burger, a steak, fries, right? We're going to start with soup, our nutrient imbalance and our fluid balance before we can get into restoring the depleted tissue. So our first steps would be to restore the nutrients and the fluids, and then secondarily, we will be providing food that is high in kilocalories, so nutritionally dense food, and high quality proteins. And lastly, we'll transition to normal food intake and introduction of physical activity. So let's talk about vegetarian diets for a moment. As I mentioned, people with vegetarian diets have to be careful that they're eating complementary protein sets. But people choose vegetarian diets for several reasons: for environmental reasons, because it reduces their global carbon footprint; for health reasons, because they want to eliminate fats like butter and animal fats; for ethical or religious reasons, right? They just don't feel good about consuming animals. But vegetarians must be very careful to consume adequate amounts of multiple different types of foods and need to plan their meals out very well to make sure that they are meeting not only their caloric but also their nutritional needs. Now, vegetarians come in many shapes and forms. We have lacto-vegetarian, which is going to eat grains, vegetables, fruits, legumes, seeds, nuts, and dairy foods. So they allow dairy, but they avoid meat, fish, poultry, and eggs. Lacto-ovo will be able to eat dairy and eggs, so grain, vegetables, fruits, legumes, seeds, nuts, dairy foods, and eggs, but still avoid meat, fish, and poultry. Now, an ovo-vegetarian will eliminate dairy foods but continue to eat eggs, and a vegan will eliminate dairy foods and eggs and any types of animal products, including things like honey, which is considered an animal product. Now, a semi-vegetarian can kind of flip back and forth between eating meat, fish, and poultry on occasion, but mainly focuses on diets that are going to be heavy in grains, vegetables, fruits, etc. So what is the benefit of a healthy vegetarian diet? And notice the word healthy here, right? You could literally just eat nothing but potato chips and it would be considered a vegetarian diet, but not a healthy one. Healthy means that it's going to be well balanced in all of the major food groups and get the kcals and all the nutritional aspects that you need in a diet. But if we're able to maintain a healthy vegetarian diet, we can reduce our risk of heart disease, diabetes, high blood pressure, cancer, stroke, obesity, and many more. So actually, a healthy vegetarian diet can be something that is very beneficial to the individual, particularly if they are already at risk of one of these major disorders. However, individuals who are in a vegetarian diet and are not closely monitoring what they are consuming end up at risk for low intakes of key nutrients, including proteins. Remember, we have to make sure we take complementary protein sets to make sure we get all of our essential amino acids in. Calcium, this is going to come from dairy products, so we have to make sure that if we're not eating dairy products, we get it from exogenous sources. Iron, vitamin B12, zinc, vitamin A, vitamin D, and omega-3 fatty acids, which are generally going to come from fish. So, how are we going to get these major nutrients from a vegetarian diet? Okay, so protein, since we're not getting it from animal sources, have to come from a combination of soy foods, legumes, nuts, and seeds. And again, it's going to have to be complementary to make sure that we're getting all of our essential amino acids in. The nutrients calcium can come from calcium fortified foods such as soy milk, orange juice, and tofu, so you'd be taking these basically exogenously. But we can also get lots of calcium from our vegetables, our leafy greens like bok choy, broccoli, kale, collard greens, and okra. With iron, again, we're going to typically get this from iron-fortified foods such as cereals, enriched grains, pastas, etc. Vitamin B12, again, exogenous. We're going to get that from fortified foods such as cereals, soy products, yeast, and yeast extracts. Zinc is something we're going to be able to get from soybeans. So as you see, soy is going to have a lot of what we need in terms of the complements for individuals who are not getting meat proteins. So very oftentimes vegetarian products are going to be soybean based. Omega-3 fatty acids, again, that's going to come from walnuts, flaxseed oil, and soybean and canola oil, when previously it was coming from fish and fish oils. So if we're eliminating fish from our diet, we're going to have to get our omega-3 fatty acids somewhere. And vitamins A and D can come from foods high in beta-carotene, like spinach and carrots. And from vitamin D, again, this is generally going to be fortified milk, so fortified products. But soy products also provide vitamin D. Ready-to-eat cereals and yogurts can also provide vitamin D. Typically, again, though, it's going to be fortified or strengthened with these biomolecules or nutrients. Okay, so if we're having a healthy vegetarian diet, we want to make sure that we get the major food groups in: 6 servings of grains, 5 servings of legumes, nuts, and other protein-rich foods, 4 servings of vegetables, 2 of fruits, and 2 of fats. Now, we want to not only make sure that we're getting in those food groups, but that we're getting calcium-rich food groups, because calcium is something you can quickly become deficient in, especially if you're not getting in your dairy products. So you want to make sure that you're getting the calcium from your whole grains and from fortified cereals, from your leafy greens and vegetables, as well as fortified fruit juices, etc. So thank you guys so very much for bearing with me today. I really appreciate you coming to my lecture, and I will see you guys next time when we talk about vitamins in Chapter 7. Thanks so much, guys. Have a great day and happy studying.